And now for something really different. I'm not a space scientist, like all of these gentlemen and ladies who've been up here. My only claim to space science is I've conducted some missions to analog stations in the Arctic, but that was as an amateur. I'm reminded, however, because I do give talks to children about space, and I was giving a talk to a group of first graders, and I asked the question, what's the largest planet in the solar system? And this little girl raised her hand feverishly, and she was terribly cute, so I said yes, and she said, I don't know. And I said, that's okay. And she said, but. And I said, but what? And she said, I like salmon. I said, well, that's nice. And then she said, and? And I said, and? She said, salmon is brain food. Good. Does anybody know what the largest planet in the solar system is? And she said, so, and I was a little angry. I said, so what? She said, I'll bet I can figure it out. And I thought about that for a moment. And I said, if I tell you it starts with J, she said, Jupiter. I said, you figured it out. So in a minute, when you start to ask questions, knowing I'm not a space scientist, and I say I like salmon, you'll understand what I'm talking about. The questions can be very important. Hello? There we go. In fact, a very famous question was posed by his arch nemesis Lucy to Pigpen. And she asked him, why are you always so dirty? And he responded, rhetorically, basically, who am I to disturb the dust of countless ages, was his answer. Actually, that's a very important question to us as we sit here. It's certainly a very important question to me and my company. What we do, and what I do have expertise in, is taking biological molecules that are doing something really cool in nature, and in training those biological molecules in surfaces, and asking them to do the very same thing they're doing in nature in that surface. There are, as you may suspect, a giant number of interior coatings on spacecraft. In fact, ladies and gentlemen, I think we have to ask the question, rhetorically, who are we, in fact, to disturb the human microbial ecosystems of countless ages? Because we may be doing so at our peril. And that's what I'm going to talk to you about today. This isn't just important for us in the space community. In fact, we are all pig pens, ladies and gentlemen. The Human Microbiome Project was started in 2008 by the National Institutes of Health to study this very issue. Because frankly, we really don't know much about the microbial ecosystems on our own bodies. What we do know is the total number of microbial cells on your bodies outnumber the total number of human cells in your bodies by a factor of at least 10 to 1. And perhaps more disturbingly, especially in the age of MRSA and even worse towards microorganisms, is that the total number of genes that are associated with the human microbiome may exceed by a total number of the human genes, in other words, microbial genes to human genes, by a factor of at least 100 to 1. Now you take those same genes and those same microbes and you stick them, as Dr. Becker said, in a stressful situation where there is excess radiation, there is microgravity, there are all sorts of other environmental changes, and they will respond, and they live on you. Now, there are a plethora of microorganisms. We have yet to discover them all, just on our own bodies. We do not know very much about their genetic systems. We do not know very much about, in fact, how they interact with one another. But what we do know is that they exchange genes frequently. They give them to the share. Okay? That's, in fact, how we start to see development of antibiotic resistance and these kinds of things. So we have to be cautious about how we approach these problems. Now, cleaning closed environments is problematic. It's a challenge. You're not going to have the luxury, as we've heard many times, especially as we relate to long duration missions, of routinely airing it out. And you're certainly not going to be able to use a lot of toxic, caustic, or even merely noxious materials to clean. Antimicrobial substances in particular that we use cannot promote the development of resistance to microorganisms because of that will be a long-term problem, perhaps even a lethal problem. The best approach, in our opinion, and I think that this is probably shared by a lot of different thinkers, is that we need to use a dynamic cleaning system. We need something that we can prophylactically place in the closed environment that can be tuned to harmonize with this human ecosystem that we carry around pig-pan fashion on all of us. To the extent possible, what we try to do is to harness natural molecules to, in fact, achieve this natural balance. And I'm going to show you what I'm talking about. By the way, we have just begun this. What you're seeing is some of the very first functional coatings. We call them functional coatings. You're seeing some of the very first ones that are there. What I hope today to tell you is that these kinds of things are very soon going to be in your toolbox. They'll be in your toolbox domestically, and they will be in the NASA toolbox. We use bio-based molecules, as I said, and we hope that we suspect that these are going to be carried in past Leo. There is virtually no surface on anywhere, but certainly on the International Space Station, that lacks a coating. And I mean that literally. Everything is coated. The walls, the floors, your clothing, my glasses, your cell phones, they're all coated in a manufacturing process where a separate layer is laid down as a coating. Now, those coatings are everywhere. They are in very high potential microbial load situations, the toilets. They're certainly in the food preparation areas. They are in the water recycling areas and systems. They are certainly in the bathing systems. They are certainly in the air ventilation systems. All of these coatings in the sleeping quarters, in the workout facilities, all of these, every single surface you see on that plethora of surfaces is coated. Now, every spacecraft surface is coated. And I said that again because I want you to think about it. What if we could turn those surfaces from a potential liability, because they harbor microorganisms, or noxious fumes, or they harbor grease, or oils, or dirt, into an asset? Because though to the naked eye these coatings look like, and they feel and look like they basically are two-dimensional, they are anything but. These coatings are three-dimensional. They have a great deal of dimensionality. They're highly porous. They exhibit high tortuosity. They have wormholes going through them. Even though they're 5 microns or 50 microns or 20 microns deep, from the standpoint of the polymer chemist, they're huge playgrounds in which to stick things. Okay? Now, the actual surface area of a coating, therefore, underneath your finger, is anything but the length and width of that fingerprint. Here's something I've asked some of my scientists to do. I asked them to take a typical spacecraft interior coating. This one is a 2-part polyurethane at a 1x magnification. That surface would feel just like this surface that's in front of me on this podium. I asked them to paint it on a block of plastic that I could cut. Then I asked them to give me an 800x view of that coating. And you can start to see all of the nooks, all of the crannies, all the hills, all the valleys in that coating. Now that is a coating that feels monolithic to my touch. Okay? Then I asked them to take that same coating, flip it on its side, use a microtome and cut through it, and expose it, and use a scanning electron micrograph to look at it. That is the coating, right? This is the layer that it was coated upon, and that's the coating itself. And you can see tremendous amounts of area within this coating. Lots of pores, lots of tortuosity. You can't really see the tortuosity, but trust me that those pores are not just straight down. They bend through the coating just like this. There's tremendous amounts of area where you place pigments in case of a pigmented paint. Other kinds of things that cause whitening, like titanium dioxide and zinc phosphate, can also be placed in these coatings. Now, if such a huge amount of surface area was put to work on a spacecraft, what would we get? Now, traditionally, coatings have protected the underlying surface. In some cases, they've decorated the underlying surface. And what I'm here to tell you today is that there's the advent of a new criterion, and that is functionality. Not to brag, And this is really just to tell you that the coatings industry has adopted this. They understand it's the next criterion to be used in coatings. But we received the 2008 American Coatings Award for innovation for functionalized coatings. They understand it, and they're working on it right now in laboratories as we speak. You are going to have an expectation that every coating you touch within 10 years will be functionalized. It will do something dynamically in your environment to your benefit. Here's an example of one. Here's an enzymatic self-cleaning coating. Now in this case, we've got an acrylic latex, and there's one panel, and it's painted. Oops. There's painted on one side with a acrylic latex, either case. This one does not. The left-hand one does not contain an enzyme called lipase. The right one does contain an enzyme called lipase. And what you can see, if we crab it out with your kitchen's worst nightmare of soy oil, cooking oil, then over a course of time, that coating without any cleaning, dynamically eliminates the oil from that surface. Here's another example. Well, I'm sorry. Obviously, there's lots of uses for those kinds of coatings on a spacecraft or on a domestic transportation vehicle. You know, you've got optics you want to get cleaned and degreased. You've got electronic display screens all over the place you want to be degreased. Textile, processed water, HVAC systems, all these things would benefit by the self-cleaning capability, so you do not have to introduce caustic cleaners into the system. Here's another one. Now, this one probably doesn't have a Marco G environment analog, because this deals with getting rid of pesticides on surfaces. Frankly, I think roaches will follow us into space, and let's hope we don't have to use pesticides, but if we do, here's a coating that on one side has no additive. In this case, this is an organophosphorus hydrolase enzyme. On the other side, there is such an enzyme, and right here we've trapped hapless fruit flies, about 100 of them under petri dish tops, after we have toxified this surface with a pesticide. And we allow time to transpire. Hopefully this will run. And what you'll see over on the left-hand side, that the flies that have the additive in their surface are protected. Those that are on the right-hand side with no additive die like flies, no pun intended. And it's a very dynamic. This has you might suspect, some military applications, and the military is looking at this now. So those were enzymes. I don't have a dynamic video for you to show you this, but here are some natural peptide biocides. This is actually at the University of Southern Mississippi. They conducted research for me where they painted fence panels with biocides that were made out of peptides, two different coatings. The left-hand dirty coating does not contain the biocide, the right hand does. And after 18 months of study, you can see that this basically looks like the day it was painted, whereas there's a tremendous amount of microbial contamination of this. Now remember, folks, that peptide, well, you don't know this, but that peptide is 7 amino acids long. You can place it in your smoothie and get a protein boost. You can eat it. Okay? It's not toxic in any way, form, or fashion, except to microbes. The difference being that we have different cellular cell wall systems, or cell membrane systems. I'll leave you with this. Basically, there are a tremendous number of enzymatic functionalities, antibody functionalities, receptor functionalities, peptide functionalities that we can extract from nature, place into coatings in the ISS that will dynamically do things to the benefit of the crew. We do not have to use toxic biocides. We do not have to clean with caustic chemicals. We can utilize this in our toolbox. And there are a tremendous number of functionalities. Self-cleaning additives with proteases, deodorizing additives with aromatic hydrolases, all sorts of things, ureases, halogenases, anti-allergenic molecules and the like. So we think that the functional coatings will be there to help us get there. We think they'll help us explore there. We are confident they will help us live there. And we believe that eventually, if we have to, and we have to back out, that they will help us actually leave there. Thank you very much.